I think we'll start. Hello, everyone. Welcome to the Connect Science webinar. On the slide in front of you, you can see the website where you can look up our schedule and find recordings of our seminars. Today, I'm very pleased to welcome Ming Li from Sloan Kettering. Ming is an immunologist at Sloan Kettering and a professor at Weill Cornell Medical Center. He went to Fudan University in China, got his PhD from Columbia, and was a postdoc with Richard Flavell at Yale before joining Sloan Kettering about 16 years ago. His research has been very effective at studying how T-cell immunity is regulated, especially in response to cancer. He's studied roles for TGF-beta in T-cell differentiation. He's identified a Warburg effect or dependence on glycolysis of activated T cells, and has studied also the role of tumor-associated macrophages in regulating T lymphocytes in the tumor. For a few years now, he identified a subset of innate-like lymphocytes in tumors. And today, I believe he's going to tell us more about that subset and its role in immune defense against epithelial cancers. So Ming, we look forward to your talk.
Great. Thank you, Judy, for the invitation and the introduction. It's indeed a great pleasure to be able to participate in this great online seminar series.
Let me interrupt you because I forgot. When you have questions, it would be great if you could put them in the Q&A, and we'll try to get to as many as we can in the question period. Thank you. Sorry.
Sure. So as Judy introduced, so my lab is generally interested in understanding immune regulation mechanisms, not only in a steady state, but also under various challenging conditions, including cancer. So for today, I will just share with you some of our recent work on how the immune system may be elicited as a defense system against epithelial cancer. Now, the topic of immune system function probably most worked out in the context of how immune system respond to pathogen infection. So in this case, we know there's a very elaborated innate and adaptive mechanisms so that the specific pathogen-associated antigens can be identified, result in their clearance. And at the same time, there's a built-in control systems, mostly for the adaptive immune response, so that the self-reactive lymphocytes may not go against soft tissue, and that's the topic of immune tolerance. So cancer, in fact, is really a unique challenge to the immune system because on one hand, it's a pathological state, and thus that does make sense if some kind of immune defense mechanism against this type of pathological challenge. But on the other hand, cancer derives from healthy soft tissues, and that's how the self-tolerance response with healthy cells may also come into play in terms of to deal with the tumor challenge, the cancer challenge. Now, in terms of potential defense response of the immune system for cancer, this was probably first articulated or proposed by Paul Ehrlich in the early 1900s. So in this case, he envisioned because of the long lifespan of various mammalian species, including us, then there's likely a positive function via the immune system to defend against cell transformation. It's probably the first conceivement of the idea of immunosurveillance. Now, specific molecular cellular basis of immunosurveillance was more conceived by Frank Burnett at the time when he proposed his clonal selection theory of lymphocytes, to the point that one of the function of the adaptive immune system might be for the defense against the cancer. In this case, would be detecting potentially mutated antigens in tumor. This was in fact conceived before, for instance, immune defense against viral infection become a critical function, and thus, for the cellular immune system. At the time, Frank conceived that this perhaps can be relevant in cancer. We know to date, some of the Frank Burnett's prediction turned out to be very much validated, to the point that tumor-associated new antigens is a major driver of how the adaptive immune system will sense and respond to tumor. But yet still different from this, a purely effective response against cancer. We know there's various checkpoints built into the system, perhaps as evolved immune tolerance mechanisms against, for instance, healthy tissues, to the point that even some of the tumor antigen reactive T cells, they will not have a robust function to effectively eliminate a tumor. One of this pathway is around the regulation by this PD-1 inhibitory receptor by the respective ligand PD-L1. Now we know, in fact, this pathway can also be targeted so that the antigen-specific CD8 T cells can now be revived to regain the anti-tumor function. In fact, to the point that the therapeutic response against PD-1, PD-L1 class of drugs appears to be most effective in cancer types has highest mutation burden, right? So, with that really a breakthrough development in the oncology field, that still really poses the question, which is that for the cancer types that have relatively low mutation burden and thus may not respond effectively to this immune checkpoint blockade-based therapies, could these still be other type of immune defense responses, cancer defense responses might be targeted or for cancer immunotherapy? So that's our topic I'm going to share with you today. So really to address that question, we thought perhaps to take a glimpse into what's going on, for instance, in human tumors, in terms of this different tumor mutation burden associated immune response, would be pick a tumor type, for instance, ideally in the same tissue, but yet still with no difference in mutation burden and the response to immune checkpoint blockade therapy. So to this end, we decided to focus on kidney cancer because there's two very distinct type of kidney tumor, one being the clear cell renal cell carcinoma, in which that has relatively high mutation burden and also responded really well to anti-PD-1 or PD-L1 class of drugs. And this in contrast to this chromophilic renal cell carcinoma, which has really low mutation burden and has almost no response to immune checkpoint blockade therapy. So in collaboration with Ari Hakimi, a physician at Memorial Hospital, we started to profile immune infiltrates in chromophore and clear cell RCC patients. So in this case, we focus on this tumor immune infiltrates from one chromophore, one clear cell RCC patients. So we could uncover various lymphoid myeloid cell clusters. Initially focused on CD8 T cells, we could recover 3 different clusters. So the first cluster is defined by high expression of S1PR5, so one of the trafficking molecules that will promote circulation pattern of lymphocyte migration, as well as this transcription factor KF2, which again is known to promote expression of target genes that promote, that support blood circulation pattern of lymphocyte trafficking. So this suggests to us what represent cells that are likely passenger cells in the tumor tissue. And this in contrast to these two subclusters of cells that are defined by high expression of PD-1 and the transcription factor TOX. As we know, the PD-1 is induced following antigen stimulation, and thus these two clusters will represent likely to be antigen-induced or even perhaps new antigen-induced CD8 T cells. The difference between the A and B subclusters are essentially whether or not they have high expression of these immediate early genes, such as FOXP and alpha-1. These perhaps reflect whether these cells are acutely stimulated by the antigen in the tumor tissue. Now, if you look into the distribution of the three different classes of CD8 T cells in these two different type of RCC tumors, what really stood out is that this number 2 clusters essentially more enriched in the clear cell RCC tumor, while this number 1 cluster, which is more circulating passenger CD8 T cell population, mostly is present in the chroma RCC tumor. So we validate these findings by analyzing a few more fresh paired tissue samples from both chromophobe and clear cell RCC patients. So in this case, in line with the induced PD-1 response, which we validated in the flow cytometry experiments, we also saw expansion of CD8 T cells in the chromophobe RCC tumor compared to the adjacent kidney or the tissue. And this suggests to us, indeed, this is a antigen-driven, tumor antigen-driven response that was readily induced in the clear cell RCC patients. And this is very much in contrast to chromophobe RCC patients. We barely see much of difference for the CD8 abundance or in between the healthy kidney and the tumor tissue, and also almost very few of these tumor-associated CD8 T cell express PD-1, further confirming that the chromophobe RCC patients associated with this low mutation burden, there's not that much of antigen-specific, tumor antigen-specific T cell response to be induced. This also perhaps explains why chromophobe RCC patients do not really respond to PD-1, PD-L1 class of drugs. Then the question becomes, if there's no such new antigen-driven CD8 T cell response in chromophobe RCC patients, are there other tumor-induced response that might be interesting to further define in these tumors? So to this end, we'll focus on this innate lymphocyte cluster. These are mostly type 1 innate lymphocytes, and also we see broadly 2 major clusters. The first cluster has high expression of S1P alpha and KF2, the same 2 genes that we define the number 1 cluster of CD8s. And again, suggests it's more of a circulating pattern of these innate lymphocytes. This is mostly characteristics of more conventional natural killer cells. There's another cluster which has high expression of integrin A1, which encodes for this integrin molecule CD49A, as well as ZNF683, which is a transcription factor. Also, is called Hobbit, which are known to define this another different subset of type 1 innate lymphocytes named innate lymphoid cells. So type 1 innate lymphocytes, or in short, RIC1s. Now, if you look into the distribution of these 2 innate lymphocyte populations in chromophobe and clear cell RCC patients, what stood out is that the chromophobe RCC patients have the much more abundant this RC1 subset population in the tumor than the clear cell RCC tumor sample. So we validate this response also with flow cytometry from multiple cancer patients. Now, not only use CD49A again, which is encoded by integrin alpha-1, but CD103, which is another integrin molecule. Both of them are known to promote this tissue residency phenotype of lymphocytes to define the response. So in fact, it's quite striking. Essentially, we saw robust induction of this 49F13 double positive RC1 populations in the tumor tissue compared to the kidney or the blood. And the double negative populations will be the circulating NK cell population. This induced RC1 response was present also in the clear cell RCC samples, but to a lesser extent. So it's quantified over here. To really look into potential function of the RC1s in tumor, we further derived a gene expression signature of RC1s by looking for transcripts that are most enriched in RC1s compared to all other tumor-infiltrating leukocyte populations. So these sets of the genes included some of the expected targets such as integrin alpha-1, but also some of these interesting molecules such as granzyme A, which suggest potential cytolytic potential of these cells, which is interesting because RC1s are conventionally considered to be more helper-like innate lymphocytes, and thus will not have really granular cytotoxicity functions, but nonetheless, granzyme A seems to be expressed at high level in RC1s. Now, with this signature, now we can mine further the broad TCGA database for chromophobe RCC patients and ask the question, could patients with a high and low level of this RC1 response potentially have different clinical outcomes? So with this analysis, we could show is that essentially the patients with a higher value of the RC1 signature appear to have much better survival than the group of patients that have low signature. That really supported a probably important function for RC1 in suppressing tumor progression or tumor surveillance function of RC1s. We wish to further extend this study to other human cancer indications, and to this end, we decided to look into breast cancer patients. If we use the signature to plot over all breast cancer patients in the TCGA database, we saw a trend to be better survival at early time point, but nonetheless, that did not reach statistical significance. And then we thought perhaps like RCC patients, that the significance of this RC1 associated immunity may be more relevant for subsets of the breast cancer patients. So to this end, we use the different oncogenomic markers that can be used to subset breast cancer patients, looking for which subset might be more amenable to this RC1 type of cancer surveillance. What we come out of this analysis is that for the breast cancer patients that harbor this hotspot gain-of-function mutation for PIK3CA, for this group of patients, this RC1 signature really predict much better survival than the ones that have low of the signature. That really supported maybe perhaps there's some special oncogenomic specification of the significance of this RC1 response in breast cancer patients. Obviously, with human patient analysis, we could only do that much, mostly just association instead of, to look into causality more definitively, in which that, we have to think about potential animal models in which we can manipulate respective pathways if, consider that this type of response might be conserved from mouse to human. So to this end, we wish to look into mouse cancer models or epithelial cancer, and to ask this question, could this type of innate lymphocyte-dependent surveillance may be conserved in surveillance of murine cancer? And still, even up to this date, a good amount of effort in the field in terms of understanding immune system response to epithelial cancer will still be using these transplantable tumor models. And many of these use the cell lines developed from epithelial cancers and consider them to be possible models for the parental tumor types. But that comes with a major caveat. For instance, this is a recent study from Pfizer, really looking into a profile of the major cancer cell lines used in the immuno-oncology study. And to the point that many of these supposed to be epithelial cancer types, in fact, will already lose epithelial properties because of the passage in the cell culture. In fact, they more look like mesenchymal phenotypes. So these are looking into, for instance, catechin vimentin regulatory expression of many of these cell lines. In fact, for many of them, compared to this parental endogenous human tumor samples, they will have loss of the epithelial properties and make them to be not ideal models to understand immune responses against epithelial tumors. This also, on top of this, all these other caveats of with the transplantation tumor model will lose endogenous tumor tissue architecture that also is critical to foster a lot of this tumor immune interaction. So therefore, to this end, we decided to use other models, specific genetic cancer models, to look into this tumor immunology question. So we decided to focus on the polyamidal T model of breast cancer because the polyamidal T oncoprotein, despite its viral oncoprotein, it activates the canonical human relevant oncogenic signaling pathways, including PR3 kinase signaling pathway. Considering PR3 gain of function mutation associated with the so-called IC-associated segregation of survival, we thought this model could be perhaps reflect this PR3 gain of function mutation of breast tumor types. Indeed, this PRMT tumor model also recuperated many of the other type of human breast cancer features, particularly more voluminal type breast cancer, and also has expression of biomarkers associated with poor prognosis. So in this particular model, with single cell RNA sequencing type experiments, we could also identify two innate lymphocyte populations infiltrating to the tumor. The first cluster has high expression of S1P alpha and KF to very much to the human cluster, which is essentially represent the conventional mouse NK cell population. And this another cluster which has high expression of integrin alpha-1 and also Harbit, which has a phenotype of the IC1 subset. What's also interesting was that in human, we saw the granzyme A is the granzyme appears to be expressed at a high level in the IC1 population. But in mouse, in fact, this IC1 subset is expressed two other granzyme, granzyme B and granzyme C. And granzyme C is particularly interesting because it has almost no expression in NK cell, but has robust expression in IC1s. So I'll come back to that just in a little bit. Now, so these studies really supported some of our early observation. In fact, that's story published in 2016, in which we looked for signs of cancer immunosurveillance in this model by focusing on lymphocytes that may express high levels of granzyme B. So with that, a parameter of assessment of immune response, we could show that cell transformation, in this case, mammary tissue from PRMT transgenic mice, will have much have much expansion of this granzyme B expressing innate lymphocytes compared to healthy mammary tissue. These cells we initially defined has more of IC1 phenotype, and also very much distinct from this 49 negative granzyme B low NK cell subset. But nonetheless, associated with the high expression granzyme B, they can have potent cytolytic activity against cancer cells in the single cell killing assay. And the cytotoxic activity is indeed little granule mediated, because if we use perforin deficient cells, they will lose this cytotoxic activity. That really support the idea that these cells can indeed have little granule dependent cytolytic activity against cancer cells, despite they have more IC1 cell phenotype. And in fact, that did raise the question at the time when we report these observations, which was that the field, despite the recognition of the more expanding subsets of innate lymphocytes to the inclusion of the cells from the innate lymphocyte cell lineage, the conventional IC1s are considered to be more of a help equivalent of innate lymphocytes. They will not have cytotoxic activity, which would be distinct from NK cells. Now we reported this more tissue-resident IC1 type of cells, like cells, which express high levels of granzyme B and can have cytotoxic activity. That really raises the question: what is the origin of these cells or the ontogeny of these cells in terms of this differentiation pathway. You could imagine maybe perhaps this represents distinct lineage, or they may be converted from NK cells, or maybe perhaps this represents IC1, just like some of the cytotoxic functions overlooked in the field at the time. So we wanted to look into this question more closely by considering generating mouse models that can use to map and also do tracing experiments to looking for particular conversion between NK cells and this type of cytotoxic IC1 phenotype of cells. So to this end, we performed also ATAC sequencing experiment looking for gene loci that may be more selectively open between IC1s and the NK cell. In this case, in fact, we even focus on more canonical IC1s from the liver and comparing IC1s in the liver to the NK cells in the liver. And in fact, the Grand-C locus really stood out because not only Grand-C is selectively expressed in IC1s, but also gene locus are selectively open in IC1s, not in NK cells. That perhaps associated with really distinct differential expression of Grand-C in NK cells and IC1s. We also picked S1PR5 to be the locus target for the conventional NK cells because, again, the gene locus is selectively open in NK cells, not IC1s. So we made two knock-in reporter and the Cree mouse strains. So for the Grand-C locus, we knocked in TD tomato and also a Cree expression cassette. For the S1PR5 locus, we knocked in a GFP marker and also Cree line so that we can not only identify the cells with fluorescent proteins, but also use Cree recombinase to do cell-fate mapping experiments. So the first question we asked was that could this Grand-C expressing IC1 phenotype cells might be converted from S1PR5 expressing conventional NK cells. So to this end, we performed a fate mapping experiment in which that S1PR5 Cree can indeed map most of the conventional NK cells, in this case defined by expression CD49B, but no expression of CD49A. But importantly, this Cree does not map the cells, the IC1 phenotype cell, the 49A positive subset. Even for these IC1 phenotype cells from the salivary gland, in this case, in fact, there's two relatively phenotypically distinct populations. One expresses CXCR6, has no expression of mesodermin, the transcription factor typically used to define natural killer cells, but as well as this 49A positive humus expression by CXCR6 negative or low subset. But neither of these subsets appears to be fed mapped by S1PR5. Also suggested that even for this humus positive subset of RC1 phenotype cells, they are not converted from S1PR5 positive NK cells. Now, if they're not converted from S1PR5 positive NK cells, could they indeed differentiate along the IC lineage? To this end, we performed transfer experiments with the defined IC progenitor cells by transferring them to Rag gamma C deficient mice. We could show that, in fact, they can readily differentiate to this 49A positive Grand-C expressing cells, really supported the idea that indeed these Grand-C expressing cells can be differentiated along the IC lineage. Now, these Grand-C expressing cells, they arise quite early in mouse development. In fact, we can already see these cells in the fetal liver and also around the neonatal stage. And these are much earlier than the conventional NK cells. They appear mostly around like 2 weeks of age. And in fact, some of these NK phenotype type 1 innate lymphocytes were considered to be immature NK cells in some of the old literature, right? So that also raises the question, could these Grand-C expressing cells might be progenitor cells for conventional NK cells, right? So, we explore this possibility by performing this reciprocal reverse fate mapping studies. Now we can use the Grand-C Cree to breathe to a reporter and ask the question, could any of the Grand-C expressing cells converted to conventional NK cells? And the answer is no. In fact, almost none of these conventional NK cells, other than the spleen or liver, will be fate mapped by Grand-C Cree. that really strongly support the idea that this GRAND-MC and S1PR5 really fat mapped somewhat terminally differentiated innate lymphocyte populations, one along the IC lineage, the other along the INK lineage. So one of the questions we had at the time was that what might drive GRAND-MC expression in IC1 lineage of cells? Because with the GRAND-MC report and the fat map mouse, we can define three different subsets of the IC1s in the liver. So the first subset has a current expression GRAND-MC protein, and obviously they are fat mapped. The second subset is fat mapped by GRAND-MC Cree, but does not currently express GRAND-MC protein, perhaps reflecting a transient nature of turn on the GRAND-MC locus in the cells. And yet there's still another subset of the IC1 phenotype cells which do not express GRAND-MC protein or are fat mapped by GRAND-MC Cree. And with these three different subsets, what we could show is that not only there's regulation of GRAND-MC expression from the transition from the double negative, double positive stage, the GRAND-MC expression also appears to be upregulated in this transition. The peripheral molecule does not appear to be altered. And so therefore, this suggested more of a gaining of some of this cytolytic program along the differentiation trajectory to the state of GRAND-MC protein expression. Now, with this further subsetting, we also performed RNA sequencing experiments. In fact, this GRAND-MC expressing subset not only express more granzymes, but they also express many of the other potential effector molecules for IC1s, including some of the chemokines and also cytokines. So that suggests to us perhaps this GRAND-MC state reflects more the effector state of the IC1 lineage of cells to look into what might regulate IC effector differentiation. We looked into the possibility of I-15 as a critical regulator because I-15 is known to be essential differentiation factor for type 1 innate lymphocytes. So indeed, in an in vitro cell culture experiment, interleukin-15 and together in complex with the interleukin-15 receptor alpha chain can maintain the high expression GRAND-MC for this double positive population. But importantly, they can also induce GRAND-MC protein expression in this single positive subset and even in this double negative IC1 population in this 24-hour cell culture experiment. And this is in sharp contrast to the culture of NK cells with the same I-15 receptor complex. Essentially, NK cells, barely any of these cells will express GRAND-MC. So it still shows the specificity of the GRAND-MC upregulation, even for the I-15, to this more IC1 lineage of cells and distinct from the more conventional NK lineage of cells. So with these observations, we're also interested in defining what might drive this IC1 response in the tumor tissue. To this end, we used two different mouse models. One is interleukin-15 report mouse strain, in which we could define which cells might express interleukin-15 in the tumor microenvironment. The other is a flux strain of interleukin-15, in which we can define the function of interleukin-15 expressed in a different lineage of cells. With this analysis, we could show that despite I-15 expression in some of the hematopoietic cell lineages, including dendritic cells, even to a low extent in macrophages, interleukin-15 produced by these cells, they do not appear to be essential for the induced IC1 response in PYMT tumor. But instead, interleukin-15 appears to be highly induced in the transformed epithelial cancer cells themselves. In this case, compared to the healthy mammary epithelium, this PYMT transformed mammary cancer cell express much higher levels of interleukin-15. And that epithelial source of interleukin-15 is essential to drive this IC1 response to the point of depletion of interleukin-15 from these cells will result in much attenuated IC1 response. So that really would imply a direct cancer cell sensing mechanism of IC1 for interleukin-15, because in this case, it's important to keep in mind, interleukin-15 is typically transpresented on the cell surface by interleukin-15 receptor alpha chain, and thus, more promoting more juxtacrine signaling instead of just endocrine or paracrine signaling. So that's really supported direct sensing mechanism of ILC1s for cancer cells. To really further explore this possibility, we took advantage of this report and the query line we generated, so we can further cross this transgenic mouse line to the cadherin MCLF reporter mice, which we can use CLF to mark cancer cells, as well as this flock stop GCAMP5 reporter, because GCAMP5 essentially is a fusion molecule of GFP to calmodulin, but also this M13 peptide. But in the absence of calcium, this M13 peptide will fold back to suppress the fluorescent signal of GFP, which can be modulated by a calcium response to the point, calcium binding would abolish that inhibitory effect and thus, re-expose the GLP fluorescent signal. So that has been used extensively in the neuroscience field to look into calcium response. In neurons, we can use this in this case to look for potential calcium response in innate lymphocytes, perhaps as a reflection of immune sensing response. So we put all these alleles together and really ask the question, could indeed we can define the response in situ in the tumor? So we could define the guanine expressing cells in the tumor tissue. In fact, these cells appear to be in contact with E-cadherin positive cancer cells. Now, with this all genetically marked molecules, we can also perform intravital imaging studies to look into the mode of ILC interaction with in the tumor tissue. So in this case, I just play a short video to look into potential calcium response in these IC1 cells. Just focus on these two cells in which you can in fact see a flux of calcium during this imaging time window. In two of these cells, this cell and this other cell. What you can probably also appreciate that is these cells do not appear to migrate aggressively in the tumor tissue. They appear to be stationary in contact with epithelial cancer cells, but nonetheless, it appears to be active, reflected by calcium response. If we in fact look closely into where this calcium response was induced, in fact, much of the calcium response was triggered at the contact site with cancer cells, really strongly support the idea much of the inducing signal for the calcium response likely would be the signals or stimulating ligands from cancer cells themselves. So this really strongly suggests to us this IC1 associated cancer surveillance involved direct cancer cell interaction or direct cancer cell sensing. So this type of mode of regulation appears to be conserved in human, because if we look into where this human IC1s reside in the chromophore renal cell carcinoma tumor tissue, we could show that in fact these cells also appear to be in direct contact with cadherin positive cancer cells. And importantly, interleukin-15 likely also to be a critical modulator of this response. It is because interleukin-15 expression really tracked quite nicely with the value with the signature value RC1. And also in an in vitro culture experiment in which we can isolate the IC1 cells from tumor tissue and culture them with different doses of interleukin-15 and performing a functional assay with K56.8 as sort of a target cell as putting cytotoxic activity, which we can show is that in fact high dose of interleukin-15 is needed to promote the cytotoxic function of IC1. That collective suggests to us is that this perhaps the sensing of the tumor or cancer cell expressing interleukin-15 really a critical mechanism of whether or not this effective cancer immunosurveillance can be induced by this tissue resident or tumor resident IC1s. So that really would support a model in which we think that in addition, for instance, to this new antigen-driven type of conventional CD8 T cell dependent cytotoxic defense response, which can be suppressed by, for instance, the PD-1, PD-L1 pathway. Even for tumor types that has a low mutation burden, they can still be sensed by this innate lymphocytes, so specifically this unique subset, a unique lineage of type 1 innate lymphoid cells. One of the critical cancer cell alarming that is sensed by this IC1 cells will in fact be this cytokine interleukin-15 cell. That will in fact define very unique type of innate cancer immunosurveillance response. Okay, so that's more for the IC1 response in tumor. Now, in this 2016 story, we also reported that in addition to the IC1 cells that are induced in the tumor tissue, there are also this unique type of innate-like T cells, which are also defined by high expression granzyme B. That can be selectively expanded in the tumor tissue, but nonetheless, they have unique phenotype because, for instance, they also express NK1.1, this innate activating receptor, and they do not express PD-1. It's more of a marker to define the conventional T cells. And also importantly, these cells appear to have innate cytotoxic function because in the single cell killing experiment, they can readily kill target cells and also in a peripheral-independent manner, which is very much in contrast to the CD8 T cells, which would not kill target cells. So that really prompted us to really look into potential origin of this innate-like T cell in the tumor tissue. And to this end, we also performed single cell RNA sequencing experiments for, in this case, CD8 T cells that infiltrated the tumor. We could identify 5 different clusters. The first cluster has high expression of IL-7 receptor and TCF7, which perhaps reflect the recently activated T cells. The second cluster has high expression PD-1 and TOX, which essentially define this more of a conventional exhausted CD8 T cell subset. The third cluster has high expression NK1.1 and granzyme B. That's essentially this innate-like T cell we reported early on. Now, in fact, we call them killer innate-like T cell ITCKs. The fourth cluster has high expression of the type 1 interferon signatures. And the number 5 cluster essentially is a proliferating cell cluster. To define the potential origin of perhaps, mostly this more differentiated CD8 T cell population, we performed further this trajectory analysis and really asking the question, could perhaps this recently activated CD8 T cell be the progenitor for this more respected and more differentiated subsets? We saw a nice transition from recent active T cell to this exhausted PD-1 subset, and also including some of this more interferon signature-defined subset along this distinct transitionary trajectory. Now, for this innate-like T cell subset, we saw a less of a transition with recently activated T cells, but if anything, if indeed it occurred, it would follow very much distinct trajectory from the ones that trigger the differentiation of exhausted PD-1 population. So that really suggests to us either this differentiation pathway follow a very distinct path, or perhaps these innate-like T cells may have a very much distinct origin, may not convert from the recently activated T cells. Perhaps to really define a T cell biology, the most important is to understand is antigen specificity, right? So to this end, we decided to look into the TCR usage and antigen specificity of these 2 phenotypically distinct CD8 T cell population. So to this end, we single cell sorted this PD-1 subset and also the NK1.1 positive subset T cells from P1MT tumors, and it did a paired TCR alpha beta chain sequencing. Then with the paired TCR, we can really ask the question, could, for instance, clonal expansion occur for this respective subset? And could also, these 2 different subpopulations share the same TCRs and thus the same antigen? So with this analysis, we could show this PD-1 positive subset is very much oligoclonal, to the point the top 5 clones can make up more than 50% of all tumor infiltrating PD-1 positive CD8 T cells. And this is in contrast with innate T cell. We see some signs of clonal expansion, but it's much more polyclonal than the PD-1 positive subset. But what's also important is that essentially there's no overlap of TCR usage between this innate T cell and PD-1 subset, really suggested that they really respond to very different antigens. To really look into what could be, type of tumor antigens these T cell receptor might recognize, we used a TCR activity reporter system in which that we can express these paired TCRs that are cloned from this T cells into an NFAT-driven TCR activity reporter mouse strain. Then we can culture this reporter cells with tumor cells and looking for potential cross-reactivity with tumor-associated antigens. Now, in this case, just keep in mind, we cloned the T cell receptor from one particular PYMT tumor, but we're doing the culture experiment with the tumor lines from another PYMT tumor from different mouse, right? So the cross-reactivity would really suggest the shared tumor antigens, but no reactivity would really strongly support the idea that these perhaps react to new antigens, only unique to specific tumors, will not be shared between different tumors. So to this end, this dominant, highly clonally expanded PD-1 positive T cell, it will not react to tumors from a different T1T mice. That strongly suggests to us that, in fact, these are likely driven by new antigens, right? Very much consistent with the idea in the human that a PD-1 mostly reacts to new antigens, not self-antigens or shared tumor antigens. These are very much in contrast to these innate T cells, because in this case, almost all the TCRs, about 80% of them specifically, they will have strong cross-reactivity to the tumor, to tumor cells from the different PYMT mice. Much of this reactivity appears to be specific to the classical class 1 associated antigens. It is because if we use tumor samples from H2K1 or D1 double deficient mice, almost all of them except the one clone will lose this reactivity. That suggests to us that most of these TCRs are reactive to class 1, classical class 1 associated tumor associated cancer cell antigens. Then with this analysis, we consider two possibilities. One still potentially, perhaps this antigen is so unique, they can just drive this distinct differentiation to innate T cells, still in the context of the conventional T cell differentiation, right? So we could test this hypothesis by engineering the conventional T cell to express this paired TCR. At the same time, we can use the CRISPR-Cas9 technology to essentially knock out endogenous T cell receptors in these conventional T cells. Then we can transfer this T cells to tumor-bearing mice, ask the question, could this innate T cell TCR then really drive the differentiation of the T cell to innate T cells, right? The answer is no. In fact, this T cell receptors, in fact, would drive this differentiation to more of this PD-1 positive exhausted T cells instead of this more of NK1 positive innate T cells. That really suggests to us that perhaps the T cell receptor need to have a function at early stage, right, beyond just the triggering differentiation conventional T cells, and perhaps even drive its differentiation in the thymus. To this end, we could test the hypothesis now by putting the same TCR into hematopoietic stem cells and let it pass through the thymus to drive the T cell differentiation, and then ask the question, can now this T cell receptor drive the differentiation of T cell to differentiate to the innate T cells? And the answer is yes. Now, if we do this sort of bone marrow reconstitution experiment, have the T cell receptor expressed in the hematopoietic stem cells, now that T cell receptor can drive the T cell differentiate to this innate T cells. Not every T cell expresses NK1.1, but none of these cells will become PD-1 positive exhausted T cells in the tumor microenvironment. So in line with this more early role of a T cell receptor in driving the differentiation, then the later, mature CD8 T cell state dependent function that really strongly supported a function of the T cell receptor in drive distinct thymic selection. That's indeed to be the case, because if we look into the thymus in terms of how this T cell receptor would drive the T cell selection, we saw a very different phenotype of selected T cells. In this case, this TCR positive selected T cells, they will not express CD4 CD8 co-receptor, which is distinct from this mobile polyclonal post-selection T cell. Most of the express are the CD4 CD8 co-receptors, and thus represent conventional CD4 CD8 T cells. But yet, as you can see, in fact, even for the polyclonal T cell, there's still a portion of this post-select T cell do not express either of these co-receptors. This, in fact, is known to be the cell population are positive selected by agonist antigens in the thymus. In line with this mobile agonist antigen-associated selection, the T cell receptors that were cloned from the RTCKs from the tumor tissue, in fact, about 70% of them have cross-reactivity function against the cortical thymic epithelial cell line. That really formally demonstrates that these are indeed self-reactive T cells. And that self-reactivity really drives this distinct thymic selection that is critical for these cells to be able to differentiate to this innate-like T cell in the tumor tissue. To really further explore this idea, we further looked into the likelihood of these polyclonal progenitor cells, double negative, CD4 CD8 double negative progenitor cells, be able to differentiate to innate-like T cells in the tumor tissue. And in line with this agonist selection in the thymus, in fact, this progenitor cell express high levels of PD-1, despite the fact that the mature differentiator do not. So the progenitor cell in the thymus express, they can be defined by high PD-1 expression. And a good fraction of them express high levels of CD122, which is shared receptor for interleukin-15 and interleukin-2. We could transfer these putative progenitor cells to initially a RAC-deficient PYMT mouse recipients. So in this case, because these progenitor cells has already been shown to be the precursor for this intraepithelial REL populations. In fact, by transferring to the RAC-deficient immunodeficient PYMT mice, we could see repopulation of these cells for innate-like T cell in the tumor, but also REL type of cells in the small intestine. There's still interesting difference in terms of phenotype. For instance, NK1.1 is only selectively induced in the tumor, but not in the small intestine. We could also do this type of transfer experiments in the lymphoreplete tumor-bearing mice. In this case, we still saw sizable repopulation of these cells in the tumor tissue, but not very much in the small intestine. So this is how to quantify in terms of the difference. Now, this is consistent with the idea that the RELs are populated during very early stage of mouse development, and it is healthy small intestine tissue. They will essentially have a limited tissue niche to accommodate more of these cells along the cell lineage. And this is very much in contrast to the tumor tissue, which appears to have a growing niche in terms of accommodating more of these cells that differentiate from these thymic progenitor cells. And that is interesting observation, perhaps can be relevant for us to consider maybe target these cells for cellular therapy. Now, I already showed, in fact, this lineage of cells, not all of the cells will express NK1.1, right? Only a subset of them express NK1.1. That prompted us to also define a better, more general early marker that can be used to more broadly define this lineage of cells. So this end, we also performed RNA sequencing experiments of these innate-like T cell progenitor cells in the thymus, and looking for the transcripts that are equally induced at the progenitor stage and also the mature cells. To this end, we could identify this signaling molecule FC epsilon 1 gamma that can define the progenitor cell in the thymus, as well as more broadly define these cells in the tumor tissue. In fact, not all these CD122 FC epsilon 1 gamma double positive cells will express granzyme B or NK1.1, and thus more broadly define this lineage of cells. Very much similar to the IC1 response. This induced, the PRM tumor induced RTCK response also is dependent on interleukin-15 expressed by cancer cells, to the point that, with the cancer cell specific deletion of interleukin-15 will almost completely abolish this response in the tumor, but does not affect the thymic progenitor cells. Even a few, the cells remaining, there's a really loss of the effector differentiation phenotype defined by expression of NK1.1 or grandon B. And that is associated with accelerated tumor growth phenotype, supporting critical function for interleukin-15 expressed by cancer cells to drive this innate lymphocyte, innate light T cell dependent cancer surveillance response. But considering interleukin-15 can be a limiting factor for this response, we also explore the possibility that interleukin-15 signaling may be targeted for adoptive cell therapy. To this end, we did it, with a more genetic system, considering interleukin-15 mostly active at STAT5 to drive its downstream response. We took advantage of a considered active form of STAT5 mouse allele, which can be activated following a Cree expression. So we isolated either progenitor cells from this Cree log stop log STAT5 BCL mice, either for this alpha beta innate light T cell progenitor cells or the CD8 single positive T cells from the thymus and transferred to the PRMT tumor bearing PRMT tumor mice. Then we can give these mice tamoxifen to induce STAT5 BCL expression in this respective two populations transfer cells, and ask the question, what will happen to the conventional T cells or to the innate light T cells? What we found was that the activation of STAT5 can really robustly expand this innate light T cell in the tumor, not so very much for the conventional T cells, and much of this expanded innate light T cell, many of these expanded innate light T cell, in fact, will express high levels of grandin B and NK1.1. And this particular inducer response for the innate light T cell response can selectively suppress tumor progression, really support a unique function for this lineage of cells in terms of in response to interleukin-15 to drive this anti-tumor response in a cell therapy setting. We're also interested in defining whether this type of response might be conserved in human. To this end, we become interested in colon cancer because the early study from Zhang Zhenming's lab at Peking University, in which they could identify more IEL type of subset of CD8 T cells in the colon cancer patients. We further used Fc epsilon 1 gamma as a mark for the innate-like T cells. We could show that, in fact, in the colon cancer patients, there's expansion of this innate-like T cell compared to adjacent colon intestinal tissue. And many of these innate-like T cells, in fact, will express high levels of grandin B, very much similar to the mouse cells. This inducer response also appears to be associated with interleukin-15 expression in cancer cells, because in this case, it's a nice antibody can be used to stand for interleukin-15 expression. We could have a very nice association between the level of interleukin-15 expression expressed in cancer cells and the magnitude of this induced innate light T cell response in the specific patients that further support the conserved response between mouse or human in terms of sensing the cancer cell expressed interleukin-15. So that really support to us the model in which that this innate lymphocyte, innate light T cell response could be induced even for the cancer types that has a low mutation burden. And one of the critical sensing mechanism around the interleukin-15 expressed by these cancer cells. And to some extent, innate light T cell may also sense some of the self-antigens to further modulate its response. So with that, I want to just conclude by acknowledging lab members involved in these studies. Some of the early work was mostly performed by Saida Dadi, former postdoctoral fellow in the lab. Brianna did most of the work for the IC lineage studies. Emily did most of the work for this renal cell carcinoma probing, and James and Shen did most of the work for the RTCK studies with help from many other lab members we have also had help from many of the collaborators, both at MSKCC and in other institutes, and also various funding agencies. So I also thank you for attention. I'll be happy to take any questions.
Hi, Ming. Can you hear me?
Yes.
Okay, because I'm having a technical problem. I can't see the questions in the Q&A. If Richard is there, maybe he could help me. Can you see them, Ming?
Yeah, I can see it. In fact,
first, I'd like to ask a question. So usually cytokines like IL-15 aren't like, they drive proliferation of lymphocytes rather than being the sort of activating receptor for killing or other responses. So my question is it possible that there are other features of the tumor besides the production of IL-15 that might be recognized by the ILC1s? In particular, are there other receptors, activating receptors found on NK cells that are active? And for the killer ILTCs, is the FC receptor maybe one of those activating receptors?
Yeah, that's a great question. Indeed, these IC1 cells express many of the activating as well as activation and inhibitory receptors. And for instance, NKG2D is quite highly expressed in these cells, very much similar to NK cells. So therefore, very much possible, in addition to interleukin 15, there was sense other stress molecules as well. And we are looking into some of these pathways, for instance, NKG2D pathway, but we don't have an answer for that in terms of how essential that is. But nonetheless, what we still consider that the limiting factor would still be interleukin 15. Considering the loss of function phenotype and also even the gain of function phenotype, we could detect with STAT5CA engineering. So in other words, that there are multiple signals, like activating receptor signals, possibly cytokine interleukin 15 signal. One of the limiting factor in terms of magnitude of the response may still be because of the difference of interleukin 15 signal inside of other stress molecules.
And one other question: do you think these innate-like cells might have a function in the development of these tissues? Because you see, like in the liver or the gut, because you see them so early in. Have you knocked them out and seen any changes in normal development?
Yeah. That's also a great question, because many of these cells are considered present in quote unquote healthy tissues, right? So they are not at least not in a disease state. And what can be the function of these cells? And we are looking into some of these questions, mostly in the context of cancer than others. We have a general hypothesis. Essentially, these are the local defenders against tissue distress. And to the point that it may not have a developmental functions, it may not be essential for the developmental process, but they are mostly positioned in the tissue as the first-line defenders against any kind of many different type of tissue stress signals.
Could you look in the Q&A? There was a guy from Notre Dame who had a few questions for you, I know.
Yes.
Before I got knocked off. Yes.
I can read out the question. Yeah. Xin Lu from Notre Dame. The first question is that is S100A Cre-I15 flox mice after knocked out in myeloid cells, not epithelial cells, how did that model provide cancer-derived IL-15 essential? Yeah, so that's a great question. You know, in fact, S100A-CRE is more used to target neutrophils than other cells. But we found S100A-CRE is selectively induced in the transformed epithelial cancer cells, not in the healthy mammary epithelial cells. That's why we use this model also to target the epithelium cell lineage. Now, then the question is that how we can know is not IL-15 coming from neutrophils, but rather from epithelial cancer cells, right? Because we already used the CRE. You know, if you are interested, read our paper, the Nature Immunology paper. We use the various CREs, include even type 2 CRE that's deleting all hematopoietic lineage of cells. For IL-15, we do not see a phenotype in terms of defective IC1 response, RTCK response. So therefore, you know, by putting all this together, this really provides strong evidence that this IL-15 coming from epithelial cancer cells, not from, let's say, in this case, with S100A CRE coming from neutrophils. It's not perfect CRE. Also, by the way, we also used MMTV CRE, which will more also target epithelial cells, and to show the same phenotype. But yes, still MMTV CRE also has this caveat. It's not entirely specific to epithelial lineage cells, can also target with variance frequency in other lineage of cells. But collectively, we are confident that IL-15 produced by cancer cells will be a driver of this response. The second question from Shane is that, do you have data to show that depletion of IC1 enhanced PRMT tumor growth? Yeah, that's also a great question. The answer is yes. If you read our Science Immunology paper, in which we use the GRAND-MC CRE crossed with DTA mouse strain to select the deplete ILC, we saw accelerated tumor growth phenotype. So that's already all have been published. Thank you for these questions.
Thank you so much, Ming. I think we have to end the session. I really thank you for coming and sharing your work with us.
Sure. Thank you for having me.
Bye.
Bye-bye.